Okay, thank you. And good afternoon, everyone. It's my um, absolute pleasure to moderate today's session. I would like to start the ATI seminars with our land acknowledgements. We acknowledge that the University of Alberta is located on Treaty 6 territory and respects the histories, languages, and cultures of First Nations, Métis, Inuit, and all First People of Canada, whose presence continues to enrich our vibrant community. Following that, I would like to thank Astellas for their support of the ATI seminar series. And a quick reminder that our 2026, which is right around the corner, ATI Research Day will be held on March 25th. And we're pleased to have Dr. Kiran Kush from Stanford University as our keynote speaker. You can now register online and submit your abstract, and we encourage your trainees to present their research at this research day as well. And then it's my pleasure to introduce Dr. Mandy Ford, who is a T-cell biologist and a professor in the Department of Surgery and a scientific director at the Emory Transplant Center. Dr. Ford has a long-standing interest in regulation of immune responses by T-cell co-signaling molecules in transplantation, and her work has focused on the roles of T-cell co-stimulatory and co-inhibitory molecules in limiting alloreactive T-cell responses during transplantation. She has been NIH funded for this work since 2007, so for a long time, and has authored more than 150 publications. She has also won several awards and has served on various committees and on the NIH study section. But importantly, we just found out she is also partly Edmontonian. So thank you, Dr. Ford, for presenting today. And the seminar title is The Road to Transplantation Tolerance via Pharmacologic Modulation of Immunity. Dr. Ford, the floor is yours.
So I'm going to talk to you today about a couple of stories that my lab has been working on for a number of years regarding the use of therapeutic targeting of co-stimulatory molecules for the control of alloreactive T-cell responses in transplantation. These are my conflicts of interest to disclose. So just to get everybody on the same page, we all know that transplantation has both promise and peril. Transplant is, of course, a life-saving, it can be a life-saving treatment option for end-stage organ disease, but of course comes with the increased risk of infection and malignancy because of the need for lifelong immunosuppression. So I sort of consider infection and malignancy sort of the on-target side effects of transplant immunosuppression. There are also off-target toxicities, including nephrotoxicity and neurotoxicity that's caused by calcineurin inhibitors, the current standard of care. And even on top of these, both on and off-target toxicities, rejection is still a problem. And so I think the question facing our field as T-cell immunologists that are trying to control T-cell responses during transplantation, how can we better inhibit the immune response in order to promote tolerance? And so that's what we're going to sort of talk about today, really this challenge to design better immunosuppression to improve outcomes in our transplant recipients. And I'd like to sort of put forth that the theme of this talk is really going to be how I think the field has really evolved into this is that it's really bench to the bedside and then back again. And so to sort of highlight how we have taken what we've learned from early clinical trials and even use of co-stimulation blockers in human transplant recipients and take that information and go back to the bench to use mouse models to discover better ways to control these alloreactive T-cell responses that then can be translated. And so this is really an iterative approach to continuing to understand more about how we can better control the T-cell response during transplantation. So we all know that T cells are the orchestrators and effectors of allograft rejection, and it's really these T cells that get primed following tissue transplantation and the danger that goes along with donor organ procurement that primes the alloreactive T cell response, which then migrates back to the tissue and actually mediates rejection of the graft via effector mechanisms. And so all of our current standard of care immunosuppression, namely calcineurin inhibitors like cyclosporin and tacrolimus, are aimed at controlling this perception of signal 1 or antigen via peptide MHC recognition by alloreactive T cells. And so over the last few years, there's been a recognition that in addition to blocking signal 1 as a mechanism of controlling alloreactive T cell responses, we could also capitalize on the fact that T cells require signal 2 or costimulatory signals for their activation. And it's really this fine balance of costimulatory and co-inhibitory signals that serve to further fine-tune or tweak the alloreactive T cell response. So co-stimulation, of course, provides an extra signal that can boost the response and promote the proliferation and differentiation, cytokine production, cytotoxic function of these alloreactive T cells, as well as to promote memory formation and promote survival of these cells. So in transplantation, a number of these molecules have been targeted or attempted to be targeted therapeutically in order to control the alloreactive T cell response. And so in the talk today, I'm going to talk about two of those molecules that my lab has been working on. One is the CD28 molecule, which of course is the target of belatacept, which is currently used or at least FDA approved for use in renal transplant. And the other is the CD154 pathway or CD40 ligand CD40 interaction. So I'm going to first talk about the CD28 pathway and then talk about the CD154 pathway and how we have targeted these molecules to prevent allograft rejection. So first, the CD28 pathway. So we know that CD28 is expressed on all naive T cells. It binds to the ligands CD80 and 86, and that ligation provides a critical co-stimulatory signal that synergizes with TCR signals to activate key transcription factors that can really turn on transcription and translation and get that T cell going. And this is a critical signal for the activation of alloreactive and really all T cell responses. And so as many of you probably know, belatacept immunosuppression is it's a CTLA-4 Ig fusion protein that really binds to the CD80 and 86 receptors, ligands, and blocks ligation of the receptor CD28 that blocks that critical co-stimulatory signal. And so, as I said, belatacept has been FDA approved for use in renal transplant. There's reduced off-target toxicities, improved renal function, and reduced incidence of DSA relative to standard of care calcineurin inhibitors. However, this comes at a cost, or as was observed in the clinical trials and has really borne out as this has been rolled out into clinical practice, of an increased incidence and severity of acute rejection in belatacept-treated renal transplant recipients compared to those treated with calcineurin inhibitors. And so this was, like I said, an increased incidence, but also increased severity of these or grades of these rejections. And so my lab was really interested in understanding what are the cellular and molecular pathways that are underlying this increased observation of increased rejection using belatacept. And so one of the things that we, of course, have to keep in mind when thinking about the mechanism of action of belatacept is that belatacept blocks, it binds to CD80 and 86, right? So it's blocking the CD28 signals by binding to these ligands, which are, of course, shared with another molecule, CTLA-4. And in contrast to the co-stimulatory properties of CD28, CTLA-4 is a critical negative regulator of T cell responses. And so by blocking this shared set of ligands, you're actually in an sort of unwanted, or I think maybe at the time this was generated, it was sort of an unanticipated side effect. But the CTLA-4 negative regulatory signals are also being blocked by belatacept. And so what are these negative regulatory signals? So CTLA-4, we know, is a critical cell intrinsic negative regulator of T cell activation. There are a number of inhibitory pathways that are activated downstream of CTLA-4 signaling, like the SHIP phosphatases that can lead to down modulation of IL-2 production, apoptosis, and anergy. In addition, CTLA-4 is critical for the function of regulatory T cells as well. And so we know that CTLA-4 is able to basically steal CD8 and 86 off of APCs, and thus limiting the activation of other T cells. And so in both of these ways, CTLA-4 negative regulatory signals are really sort of the physiologic mechanisms that the immune system has evolved to inhibit the alloreactive T cell response, and we're really blocking those signals when we use belatacept. So we asked, can we re-engineer this strategy of, we know we want to block CD28, but can we do it in a way that specifically blocks CD28 signals while preserving CTLA-4 mediated negative regulatory signals? And so in this way, we went back to the mouse and asked, can we re-engineer a strategy to do this? So like I said, belatacept blocks the shared set of ligands. If you were designing this, you'd really say, I want to block only the positive regulatory event and leave intact these critical sort of the physiologic mechanisms that the immune system has evolved to keep things in check. And so we'd like to preserve these CTLA-4 mediated negative regulatory signals. So why wasn't this done before? Well, many of you may remember sort of a very tragic event in the history of co-stimulation antibodies. It was this trial where in the UK, investigators conducted a phase 1 safety study of an anti-CD28 monoclonal antibody with sort of catastrophic results, as you can observe here. And 6 healthy volunteers were underwent injection of this molecule that led to unanticipated CD28 cross-linking because this was an intact monoclonal antibody. This cross-linking resulted in sustained memory T cell activation in a way that wasn't previously anticipated in preclinical models, and a cytokine storm. This really catastrophic event put the brakes on sort of the development and translation of anti-CD28 antibody strategies across disease states for a number of years, until the following breakthrough, I guess I would say, came about. And sort of this really highlights the idea that new tools can give us new possibilities. So this was a strategy developed by a team of chemists who went on to win the Nobel Prize for Chemistry in 2018 to really develop a novel way of making specific nanobodies that can bind to a target but not have the FC portion, and therefore not have the cross-linking event that was so catastrophic in those clinical trials. And so using this technology, Bristol Myers Squibb developed an anti-CD28 domain antibody that's shown here on the right. So your conventional antibody, of course, contains numerous domains, one of which is an FC domain that can bind to FC receptors and mediate this cross-linking event that we don't want to happen. But with the specific domain antibody, it binds to CD28 and can block its ability to interact with its ligands, but not have this potential for cross-linking. And so this is was selected from a human phage display library for reactivity to CD28. It has a much shorter half-life by virtue of the fact that it doesn't have these extra domains. But we were able to test this in mouse models to determine if specific blockade of CD28, leaving CTLA-4 negative regulatory signals intact, would better inhibit the alloimmune response. So first, important to show that that use of anti-CD28 domain antibody did not elicit a cytokine storm or increased systemic cytokines in the blood. And then here are the efficacy data in terms of how this molecule stacked up against CTLA-4 Ig, or the belatacept analog, in terms of inhibiting the alloreactive T cell response. What you can see is that CTLA-4 Ig, so this is a mouse model where we're able to specifically track donor reactive CD8 positive T cells by virtue of the Thi1.1 conogenic marker. And you can see here that CTLA-4 Ig is able to inhibit the alloreactive T cell response, but that the anti-CD28 domain antibody confers increased inhibition of the response and actually confers better protection against allograft rejection in this mouse model compared to CTLA-4 Ig. In addition, we observed that treatment with the anti-CD28 domain antibody conferred better suppression of donor-specific antibody responses, as you can see here. So the CTLA-4 Ig, or belatacept analog, certainly does inhibit alloreactive DSA production, right? And this is certainly borne out in the clinical observations as well. But the anti-CD28 domain antibody actually does so in a much more profound manner. And this was true even looking out many days after skin grafting. This is a skin graft model, even after the cessation of immunosuppression in this model. So given these results, we went on to translate this into non-human primates in collaboration with Dr. Andrew Adams at Emory's Primate Research Center. This is a rhesus macaque renal transplant model. They get bilateral nephrectomies and then receive a kidney from a fully MHC mismatched donor within the colony. You can see the treatment schedule here, and similar to what we observed in our preclinical rodent model, here in this non-human primate model, we can see that treatment with monotherapy anti-CD28 domain antibody, this is lulizumab, produced by Bristol Myers Squibb, had a significant improvement in at least about half of the donors, or half the recipients, excuse me, compared to belatacept. So this really, along with no observation of elevated cytokine levels. So this really told us that specific blockade of CD28 while leaving CTLA-4 mediated negative regulatory signals intact is actually beneficial and can better suppress the alloimmune response compared to situations in which both the positive and the negative regulator are blocked. So where are we now with this? This is a sort of an evolving field, and it'll be really interesting to see where we land with this. We at Emory initiated a clinical trial using the lulizumab in renal transplantation, using the lulizumab from Bristol Myers Squibb, where we had a lulizumab treated arm versus a tacrolimus treated arm. In addition, Flavio Vincenti at UCSF also initiated a clinical trial using lulizumab. Unfortunately, the current state of things is that Bristol Myers is not pursuing the development of lulizumab at this point in time. So these are kind of on hold. However, there's another company, Veloxis, that is pursuing the development of a similar selective CD28 blocker. This is the FR104 molecule that was made by Bernard Van Hove in Nantes. And Veloxis now has this asset and is in phase 1 clinical trials for the use of this selective CD28 blocker in renal transplantation. So we will be interested to see how these trials go, and if we can really bring this to fruition in terms of providing an additional option for CD28 blockade for use in renal transplantation. I will now move to, I guess this is my summary. I think I've sort of made all these points. We think that selective CD28 blockade preserves the CTLA-4 mediated co-inhibitory signals, thus consolidating T cell anergy, exhaustion, and really promoting graft acceptance. Better preserved Tregs, better inhibits DSA. In data that I didn't have time to show, we really showed that the mechanism by which this is occurring is that selective CD28 blockers better inhibit follicular helper cells. And that's because follicular helper cells actually express a ton of CTLA4, and they're very sensitive to CTLA4 blockade that happens with belatacept. And so, just to highlight that the development of new tools create new opportunities for mechanistically based drug development in transplantation and across disease states. I will now move to discussion of the CD40 ligand pathway, or the CD40 CD154 pathway. This is another target that, of course, many investigators have worked on over many years, decades now, and another one that is still in development, and we'll be excited to see where all of this goes as well. So, as we all know, CD154 and CD40 ligand or CD1 interactions are critical for a number of different processes in immunology. Specifically, CD40 signaling through antigen-presenting cells leads to dendritic cell licensing, which is the upregulation of costimulatory molecules and MHC expression, as well as pro-inflammatory cytokine production, and has really been used, like I said, by a number of groups for co-stimulation blockade in transplantation, leading to a more tolerogenic state. And that's shown here both in non-human primate and mouse models, where either alone, this is a non-human primate model done by Alan Kirk many decades ago, or in combination with donor-specific transfusion, you can really see that blockade of the CD154 pathway can really lead to robust, durable tolerance in many different models of transplantation, including heart, kidney, and in mouse and monkey. And so this, looked like a really promising pathway for use in transplantation. Of course, there have been a number of roadblocks to the translation of CD154 therapy for clinical use in transplantation. Really, that began with the fact that early clinical trials of CD154 were halted due to thromboembolic complications. And so in these trials for both in lupus as well as kidney transplant, these thromboemboli were observed, leading to the cessation of these early clinical trials. So the field sort of then picked up and said, okay, well, if we can't specifically target CD154, and that was because of the fact that CD154 is expressed on platelets, and again, cross-linking of that antibody can lead to platelet degranulation. Can we target the other side of the pathway of this receptor-ligand interaction? Can we target the CD40 molecule and get basically blockade of this receptor ligand interaction through this mechanism? And so a number of groups did this, both in preclinical models, in rodent models, in non-human primates, and then initiated trials, clinical trials in renal transplant recipients. When we were doing these studies, early on and looking at our anti-CD154s that we had in the lab compared to the anti-CD40 antibodies, we really made the observation very, very early on that while the anti-CD40s did confer increased graft survival, right? There's definitely a difference compared to DST alone. They never seemed to work quite as well as the anti-CD154s. And this was always puzzling to us, right? Because we think of these as, two sides of the same coin. You're blocking the ligand or you're blocking the receptor. You should get the same outcome either way. And we were really seeing that clearly wasn't the case. And that was also highlighted in a meta-analysis that was published by Steve Perrin a few years ago, where he took data from a number of different labs using different antibodies. So it's not just based on that particular, the affinity of that antibody or the epitope it's recognizing, but across multiple different clones, you can really see that the anti-CD154s outperform anti-CD40s in terms of their efficacy in prolonging allograft survival. And that really was borne out in the clinical trials of anti-CD40s in renal transplantation. Insofar as these clinical trials using anti-CD40s, both in renal transplant as well as other immune-mediated diseases, really were abandoned due to lack of efficacy and failure to meet primary endpoints in these trials. So again, this, gave us the question is really why is this happening? What is the mechanism underlying the observation that anti-CD40 appears to be inferior to anti-CD154 or less able to control rejection? And so again, taking these observations from clinical trials and going back to the lab and asking, can we dissect this a little bit further? So summarizing a number of the data because they've been published here, but really what we were able to find is that lo and behold, CD154 has an alternate ligand, so a new target for CD40 ligand. So we found that CD154 is able to bind to a specific region within the I domain of CD11B. And actually, since this time, a number of other ligands have been identified, in particular the integrin class, for CD154. But we knew that CD11B was another potential ligand or receptor, really, for CD154. And so this, of course, has a lot of implications for therapeutic targeting of this pathway, right? If you have a ligand that is able to bind to two different receptors, when you're blocking that ligand, you're basically blocking both its interaction with both CD40 as well as CD11B. However, if you're targeting just the CD40 molecule, you are potentially leaving open the possibility of interaction between CD154 and CD11B. And so we wanted to ask, is this interaction between CD154 and CD11B important, or does it have a functional role in the elicitation or effector phase of alloimmunity? And so in order to ask that question, we set up a model in the lab, or an experiment in the lab, where we were able to design a peptide that specifically binds to that M7 region within the I domain of CD154. And so it specifically blocks the interaction between CD154 and CD11B, but does not impair the interaction between CD154 and CD40. And so the experiment that we set up was the following. We're using the anti-CD154 as our positive control. So that's the antibody that we know should work well. Then we treated the mice with either anti-CD40 to block this interaction, coupled with the specific peptide inhibitor called CM7 that can block the CD154 CD11B interaction, or a negative control peptide. And what we observed is the following. So the anti-CD154, of course, gives good graft survival. When you use the anti-CD40 with a scrambled peptide, you get slightly less, but statistically significantly less allograft survival. But now when we use the specific inhibitor of the CD154 CD11B interaction, we're able to bring that level back up to the graft survival that's observed with the anti-CD154. So again, showing that the CD154 CD11B interaction is important for the elicitation of alloimmunity in this model. And so just to summarize a number of the other experiments that we went on to do, again, that are published, we really found that the CD154 CD11B interaction had very little effect on the priming or expansion of alloreactive CD8 T cells in the lymph node. And so we think that really it's the CD154 CD40 interactions that are important within the node for the priming of cells. Rather, we observed that the CD154 CD11B interactions were important at the local level. They inhibited, when we administered the inhibitor, it blocked the infiltration of inflammatory cell trafficking within the graft. And so we think that this interaction, when it normally happens, is important for promoting the local upregulation of costimulatory molecules, cytokines, and inflammation within the allograft. In addition, we have evidence that the CD154 signals for driving innate immune cell infiltration into these allografts may be derived from innate immune cells as well, because in addition to the sort of canonical source of CD154 expression, which is CD4 T cells, we observe that CD154 can be expressed on dendritic cells, as I'm showing here. So there's a potential for this direct innate immune cell interaction by which CD154 provides a signal to these CD11B positive dendritic cells and really increases innate immune infiltration and activation locally within the allograft. Of course, we'd like to put forth that these results have important implications for the clinical development of anti-CD40 versus 154 blockers for use in transplantation. So how can we use this knowledge to improve outcomes? And so can we use CD154 blockers to promote transplant tolerance? Of course, we know that seminal studies done by Chris Larson and Tom Pearson here at Emory really show that there's this important synergistic effect between blockade of these two pathways that we've discussed today. So blockade of the CD28 pathway, blockade of the CD154 pathway, they really observed long-term and durable tolerance in these early rodent models. So so we want to further develop the use of 154 or the targeting of CD154 to be able to capitalize on this potential for tolerance induction. And so in order to do that, my lab has turned to a nanotherapeutic approach to specifically block CD154, CD11B interactions. And we partnered with Doctors Lily Yang and Li Zhu at Emory's Winship Cancer Center, and also in conjunction with the Georgia Tech Bioengineering Department, to create these hyaluronic acid nanoparticles that are conjugated to the specific CD154, CD11B blocking peptide. So now we have a nanoparticle that can be administered in vivo, but that is coated with this CD154, CD11B blocking peptide. We labeled it with a near-infrared dye to facilitate the tracking of nanoparticles in vivo. And I should also mention that we're really excited about this approach because, of course, we have the potential to load these nanoparticles with additional immunosuppression, additional targets to facilitate the local delivery of these targets. And this is something we're actively working on. But so we're taking this nanoparticle that's loaded with this specific peptide, and here we're administering it to murine recipients that then receive a skin graft. The recipients either received CTLA-4 Ig plus or minus an empty nanoparticle that did not contain the peptide, or the CD154, CD11B coated nanoparticle. What you can see here, and then we're doing IVUS imaging to look to see where does this nanoparticle go, or can we see it in the graft following administration. You can see that the PBS treated mice have no nanoparticle visible. The mice treated with the empty nanoparticle, you can definitely see some of the nanoparticle accumulating in the graft. However, that's much greater when we have the peptide coated nanoparticle. And that's, of course, because this peptide actually binds to that M7 part of the I domain on the CD154 molecule. And so we think that really anywhere that CD154 is increased, is upregulated, which is at sites of inflammation, aka the graft, this is where you're going to see, or we would hypothesize to see, increased accumulation of the nanoparticle, and we do. And we can quantify this over time following administration of this therapeutic nanoparticle. So again, where does it go? We see that, as I said, most of the nanoparticle accumulates within the skin graft. We really don't see appreciable accumulation within other organs like the liver, lung, spleen, draining lymph node. And on what cell type can we see this nanoparticle binding? So we certainly can see it binding to CD4 positive T cells, which we would expect because they express CD154. But interestingly, we also observe an increased binding of the peptide-coated nanoparticle on innate immune cells. So again, suggesting that these innate immune cell subsets can also express CD154 because that's the target of the peptide, and that's where we would, the mechanism by which it binds. So in terms of outcome, what happens when we treat animals with this this CD154, CD11B blocking nanoparticle? You can see the results of this shown here. So basically, these animals were treated with CTLA-4 Ig, which again is the belatacept homolog, plus or minus the peptide-coated nanoparticle. And you can see that the CTLA-4 alone treated animals, both either just alone or with the empty nanoparticle shown in yellow, have some prolongation in graft survival, but that is statistically significantly increased when we treat animals with CTLA-4 plus the specific CD154, CD11b blocking nanoparticle. When we look at the alloreactive T cell response, again, we can see that the addition of this CD154, CD11b blocking nanoparticle significantly reduces the alloreactive T cell response. And again, that's measured by looking at donor-reactive CD8 positive T cells by virtue of this congenic marker. And then the summary data are shown here that we see significant reduction in both the CD8 and CD4 positive donor-specific CD8 T cell response following the administration of this specific CD154, CD11b blocking nanoparticle. We then went on to ask what happens. So if we have these animals that are experiencing long-term graft survival, following administration of this nanoparticle, can we test for tolerance? Is there a tolerant state that is induced in these animals? And so what we did here was to re-challenge, give these animals a secondary re-challenge with the same donor type graft for animals that had surviving grafts. And so what you can see here is that, and this is importantly, in the absence of any further immunosuppression. So in this graft, they're not getting immunosuppression at the time we give them the graft. But you can see that animals that previously received CTLA-4 plus the specific CD154, CD11b blocking nanoparticle, that 8 out of 10 of these animals were able to tolerate secondary re-challenge with a same donor type graft in the absence of any immunosuppression, suggesting that in some animals, there's the potential for tolerance induction, again, by via the targeting of both the CD28 as well as the CD11b, CD154 interaction. So what to summarize what we think we've done here with these last set of experiments, what we think we're doing is that by treating animals with the CD154, CD11b blocker, we're inhibiting that local innate immune infiltration or activation. And so we think these signals are really important for that innate immune activation, and by blocking them, we're hitting the innate immune system. We know that by targeting CD28, we're blocking the priming of the adaptive immune system, both the T cell response as well as the humoral response in the draining lymph node. And so really the sort of hypothesis about what underlies the synergy between these two pathways is that by simultaneously inhibiting both the innate and the adaptive immune response via these two different pathways, that is the mechanistic basis for the synergy that we observe, and potentially can induce tolerance in some of these recipients. So where are we going from here? We now have a U19 funded through NIAID as part of the Non-Human Primate Transplant Tolerance Consortium through the NIH to test this approach in non-human primates. We're currently working on administering the CD154, CD11b blocking nanoparticle in a non-human primate model of renal transplantation. And again, the goal is testing whether blockade of this specific CD154, CD11b interaction can combine with belatacept to result in the induction of tolerance. And so to conclude, what I hope I've conveyed to you today is that therapeutically targeting CD154 may have mechanistic advantages over targeting CD40. And really, we think that the local pro-inflammatory effect of CD154, CD11b interactions, which are not blocked by anti-CD40, is what is the advantage of using of this blocker. In addition, we've shown that the ability of anti-CD154 to result in the differentiation of graft-specific FOXP3 positive Tregs and data that I didn't have time to talk about today. We think that, of course, these results may have important implications implications for the clinical development of anti-CD40 versus anti-CD154 reagents for use in clinical transplantation. And mechanistic understanding of how CD154 blockers are really working will allow the rational design of novel pharmacologic approaches and protocols to promote transplantation tolerance and improve outcomes for transplant recipients. And with that, I will end. I'd like to thank the people that have done the work, really those in my lab, as well as a number of collaborators, both at Emory and at other institutions, and thank our funders, and thank you for your attention.
Thank you, Dr. Ford, for your presentation. That was wonderful work and a lot of information. Dr. Halloran, I see your hand raised. You're still muted, Dr. Halloran.
I loved your presentation, Dr. Ford, and I congratulate you on being one of the few laboratories, probably the leading laboratory, in pursuing this line of investigation, which we critically need. In the belatacept studies, the early rejection probably was influenced by the CTLA-4 problem, which you outlined, but I have always thought that it's the legacy that we older primates have of primed cells.
Yeah.
The lymphocyte population against alloantigens, I think Robert Leckler showed a long time ago, was about 50% memory.
Yes.
And the mystery of the role of memory T cells in graft rejection continues to bother me. We haven't seen much evidence of it, and of course, it can't really be studied in mice, or probably not well in non-human primates either. Mm-hmm. So what are your thoughts about how we're going to address this?
Yeah. Wonderful question. Totally agree with you that this is a very key aspect of really, understanding this pathway, but may have implications for all of the testing of all pharmacologic inhibitors or all, molecular pathways in transplant, because I think it is a contributor to rejection in humans, and one that is not very well modeled in preclinical models. So I guess I would start by saying we have taken some steps to better model this, the presence of memory T cells in mice in two ways. One is by creating mice that have more memory T cells. So you're totally right that if you look in laboratory animals that are kept in, sterile, even the non-sterile, but the relatively, specific pathogen-free animals have only about from 2 to 10% memory T cells, whereas as you stated, an adult human has about 50% memory T cells. And due to the inherent cross-reactivity of the TCR, many of those can cross-react with alloantigens, even if that patient has never had a transplant, had never had blood transfusion, never been pregnant. There's still the potential for cross-reactivity just based on the sheer number of alloreactive T cell clones, and then a subset of those might be cross-reactive with any given pathogen, or microbe. So we have attempted to make what we call memory mice by just taking mice and infecting them with a bunch of different viruses and bacteria that we know are cleared, and then they have a component of memory T cells. And so what we're doing is testing our therapeutics in those animals to be able to really test that important parameter in our models. The other thing I was going to mention, though, specifically as it relates to this pathway, I think you're totally right. Memory T cells are really important in terms of rejection in belatacept-treated patients, but it may also have to do with the expression of CTLA-4. So we've shown that the kinetics of CTLA-4 upregulation on memory T cells is very different than on naive T cells. So when they see antigen, memory T cells upregulate CTLA-4 within like 2 hours, where it takes naive T cells 2 days. And so by virtue of that, we think that CTLA-4, the maintenance of those negative regulatory signals is going to have, and if you take them away, it's going to have a greater effect on memory T cells than it will on naive T cells. So I think it's a really important point. I think the field needs to take into account immune memory, when we are modeling immune responses in preclinical models.
And the other issue which you raised is the lack of pharmaceutical industry interest in our field. Now, I'm sure you have been advocating for us with industry, and how do we get them back in the building? Because they all left and went into inflammatory bowel disease and cancer, etc. Because it's easier to make money in those fields. How are we going to get them back? You need industrial investment.
Right. That is a great question. I wish I, of course, had an answer for that. But so this is going to turn into a promo for our session at CIOT. So we have the Cutting Edge Organ Transplantation sponsored by the American Society of Transplantation coming up in February. And I am organizing a session called The Future of Transplantation, where we're discussing specifically that topic. How do we reinvigorate pharma interest in our field? And, I feel like it's not, so I think there are things that can be done, but I think they have to be done at the FDA level in terms of having surrogate endpoints that can give a pharmaceutical company a reasonable time frame in terms of making a go no go decision on a drug. And it can't be that we're looking for changes in outcome in 10 years or 20 years. There has to be something that we can hang our hat on in a nearer time. So we're actually, as far as like what can be done, we're trying to get both FDA individuals from the FDA, as well as from a representative of U.S. Congress, to come and hear our thoughts about this issue. And because we know that patients want change. Especially someone in research and transplantation, it's sort of heartbreaking to hear from patients who say, I've been on the same drugs are being offered now that they were 30 years ago, 40 years ago. And so you guys aren't doing anything. And it's like, you want to say like, we're doing something. It's like a duck, swimming, but you can't see the legs underwater, because there's really been no substantive change, or very little substantive change in the field. So anybody that wants to come to that meeting and have your voice heard, and hopefully there'll be some individuals there to listen to us. Thank you.
Anybody else have a question for Dr. Ford? Please feel free to raise your hand. Simon.
Yes, thank you. I just had one brief question, and you may have mentioned that. I'm sorry, I joined a bit late.
Yeah.
The CD28 antibodies were the ones that caused that cytokine storm episode in the phase 1 trials in humans after not having shown anything like that in the preclinical trials. Is something different with your new CD28 antibodies that you're not worried that could happen again?
Yeah, so great question. Well, the main thing that that is different, yes, there's a big difference in that it does not have an FC portion of the antibody. So it was developed in a different way, such that it's a single domain that binds to CD28, but doesn't have the business end of an antibody molecule that can mediate FC cross-linking. But I think so that particular problem, we think as a field, the field has, been able to circumvent. But I think, are there other issues that might come up? And so I think as a field, we need to learn from that trial, and what, in terms of just anticipating other events, and really, what happened in that trial is it was tested in mice. It was not tested very much, meaning a very low N, in non-human primates. The in vitro data was weighed very heavily in terms of, or prominently in terms of the regulator's decision to be able to move forward. And so, I know we have a lot of discussions right now, in the field with, can we use other in silico models, or in vitro to take away the need for animal testing, both for mice and non-human primates. And I think this is an example of where that was sort of done and done with catastrophic consequences. And so it may be important to remember that when we think of going away from just a very traditional animal model for preclinical testing. Thanks for bringing that up. It's a great point.
Are there any other questions for Dr. Ford? You also feel free to type it into the little chat box if microphone, or if you feel shy asking a question out loud. I do have one question though, because I have actually many questions, but the one that I would like to point out is, you saw in your survival curves when you're combining these, costimulatory blockers, a prolonged graft survival, or less, or an increase in graft survival, but it was still not 100%, and that kind of made me think like, there's probably another pathway that our immune system smartly developed to make sure that to bypass another way. So, what are your other targets that you're eyeing to see what would be next?
Yeah, it's a great question. I mean, I think the really intriguing thing about those observations, just speaking generally, is the fact that we are using genetically identical animals, getting genetically identical grafts, and treated with the same treatment. And how is it possible that we get long-term survival and even tolerance in some animals, and not very little or some prolongation, but very minimal in others? I think that's one of the great mysteries of the immune system, and the fact that VDJ recombination is stochastic. All of these animals have different repertoires, and it really shows us that it's very complex. And by virtue of having these different repertoires, I mean, that's the only thing I think that makes sense, right? But that T cells are going to be slightly different. Their affinities might be different. They're recognizing different epitopes, and that leads to different outcomes. And so, we're working on a number of pathways, but I think that, I guess the way I'd answer that question is, it really highlights, I think, the need for personalized medicine and personalized therapy in transplant and probably all immune-mediated diseases. Because even when in that example, right, where we're controlling absolutely everything, it still doesn't always have the same outcome. And so we're going to have better metrics of reading out, how an immune response is progressing, looking within that specific individual, and then targeting it as necessary, whether it's blocking costimulatory molecules. My lab has turned towards looking at a number of co-inhibitory molecules and that actually goes along with the memory question, that sometimes memory T cells don't really need co-stimulation to get off the ground, but they are very reliant on these co-inhibitory pathways that allow them to put on the brakes. And so if we can agonize those co-inhibitory pathways, including PD-1, TIGIT, we might be able to better control those memory T cells. So it's a great question, and one that is always perplexing when we get that kind of response.
We're always outsmarted by the immune system, I feel like, which is good.
I agree.
In general,
I agree.
That's a good thing. But yeah,
right. It's job security, and it makes things always interesting, right? Never a dull, never dull.
Absolutely. Are there any other questions for Dr. Ford? I don't see any hands raised, and I don't see any other questions. Okay, with that, Dr. Ford, thank you very much for your presentation and your insight on this topic.
Thank you for having me. Thanks for the invitation, and thanks everyone for listening.